All right, we can go ahead and get started. Welcome to another week of Soul Seminar Online. Today, I have the honor of introducing Dr. Helen Tai. Dr. Helen Tai is a potato genetics and genomics researcher scientist at the Agriculture and Agri-Food Canada in Fredericton, New Brunswick, Canada. She received her BS in biochemistry and MS in the anatomy at the University of Toronto. She went on to work at the University of Ottawa for her doctoral studies in the fellow in mammalian genetics. Dr. Tai made an enlightened switch to plant biology during her postdoctoral fellowship and joined the Agriculture and Agri-Food Canada in 2006. Her research focuses on understanding genetic variation, underlying traits in potatoes and Solanum wild relatives using genomics, transcriptomics, and metabolomic approaches. She applies her research to potato germplasm enhancement and breeding. Dr. Tai's research covers the Colorado potato beetle resistance and the vermintimic wilt resistance. Also looking at the efficiency and cold storage quality. Dr. Tai is currently the president of the Potato Association of America and the Solanaceae Genomic Center for Sol Chair. And with that, you can go ahead and get started sharing your screen.
Hello, everyone. First of all, I'd like to thank the organizers for inviting me to give this seminar. And I really want to commend them on the entire seminar series. I've really enjoyed attending the other sessions, and it's really a great thing to bring the Solanaceae community together in this way. So I want to start my presentation off with a land acknowledgement. This presentation is being delivered from the traditional territory of the Wolastoqiyik Indigenous people of Canada. So Leptinotarsa decemlineata is commonly known as a Colorado potato beetle. In my talk, I'll also be referring to it as CPB. The decemlineata part of the name refers to the 10 stripes on the adult beetle, and it actually makes it for a fairly beautiful beetle. And I do make jewelry out of the beetle as a hobby. So, you know, it's because I have really an abundant supply of the beetle as I live in a potato growing region, and the Colorado potato beetle is a devastating pest for potato. All larval stages feed on the potato plant, and so it is considered a major potato pest. It's actually a foliar herbivore that's a specialist on Solanaceae foliage, and it actually feeds on other Solanaceae as well, but it's become a pest for potato based on the compatibility of its life cycle with that of the potato. So the Colorado potato beetle is native to Mexico. It is a major potato pest, but it's not found at the geographical origin of potato in the Andes. The original host for Colorado potato beetle is Solanum rostratum, which grows in the central highlands of Mexico. Somewhere around the early 1800s, the Solanum rostratum and the beetle made a migration northwards and was first reported in the United States in 1811. Then very quickly, the beetle made its way across the United States. It was reported in the central part of the US at the Iowa-Nebraska border in 1819 to 1920. And then the first outbreak on cultivated potato was reported in 1859 in Nebraska. Then there was this major infestation of potato in 1864. And then this infestation led to the naming of the beetle as the Colorado potato beetle, although its origin is not Colorado. It was reported in Ohio in 1869, followed by reports in Maine in 1872 and northwards in Canada in 1872 as well, in the region where I live in. And then the beetle made its way westwards in Canada and being reported on the west coast of Canada in 1919. In 1872 was the first report of the beetle in England. This was on a shipment of potato, and similarly, reports were filed in Germany in 1877 and Poland in 1878. And critically, in 1922, it was discovered that the beetle had established itself in France. And I don't blame the beetle. I mean, you know, the joie de vivre is certainly a big attraction there. And the beetle does have a life cycle that can lead to an establishment. And the life cycle that it has is that it lives its feeding life above ground, feeding on the foliage, but it will burrow underground to pupate and emerge again and feed above ground. And then in the fall, it will burrow underground and stay there for the winter to protect itself from the cold. And so once the beetle establishes this above ground, below ground life cycle, it's considered to be established. And this is what was found in France in 1922. And earlier in the seminar series, Dr. Szymanski had reported on how beetles were even thought to be a biological weapon because they have such incredible infestations during the Second World War as it was moving across Europe. So the beetle did make its continued movement across Europe, made it to Soviet Union in 1949, and then Eastern Europe in Kazakhstan in the 1950s. And by 1993, it was detected in Xinjiang, China. So the Colorado potato beetle has more or less followed potato all the way around the world. So there are a number of Solanaceae species that are acceptable to the beetle. The acceptability is when the beetle can visit the plant and stay on the plant, but not all of the Solanaceae species support the growth of the beetle. And that's important as well for the beetle to have this kind of compatibility. So it's really the Solanum species where the beetle can get acceptability, and have supported their growth. And Solanum tuberosum, potato, is of course one of the major crop plants which the beetle can infest, but it can also feed on eggplant and tomato. So there is a species range, but the plant species preference for Colorado potato beetle has become potato. So for that reason, there's been a lot of attention on the wild Solanum species in the subsection Pitota, which can intercross with potato. And potato has a little over 100 wild species that it can intercross with. And several of them have been examined for resistance to the Colorado potato beetle. And they're listed here in this table. And a lot of this work has been done by breeders all over the world, including work by a former entomologist here at the Agriculture and Agri-Food Canada Research Center. The resistance factors are variable. There are trichomes, foliar chemicals that are both volatile and non-volatiles, and other kinds of toxic compounds that are produced by the plant. And most all of the wild species that carry Colorado potato beetle resistance, and actually most of the wild Solanum that can intercross with potato overall, are diploids. However, potato is a tetraploid. So there's this sort of ploidy incompatibility that presents a challenge for introgression breeding for potato. And I'll touch on that a little bit more. The mechanisms of resistance are both antizoonosis, where the host plant produces something that causes a change in the pest's behavior. And there's also antibiosis, where the plant produces something that is changing the insect's life cycle. And these two terms can often be somewhat confusing, because sometimes a change in the behavior can actually lead to a change in the life history. The other sort of mechanism of resistance is tolerance, and that is a quick recovery from feeding injury or a life cycle adaptation for the plant where it can recover better. The plant also has inducible resistance, constitutive resistance, direct resistance, and indirect resistance. In inducible resistance, the insect triggers the resistance mechanism in the plant. In constitutive resistance, the plant is making an investment of resources and energy into producing a resistance mechanism that's always there, regardless of whether or not the pest is present. In direct resistance, the plant directly affects the insect, either through producing some compound or some kind of a resistance barrier of some sort. In indirect resistance, the plant is producing something that attracts another organism of some sort. Either this is a parasitoid, or it could also be a pathogen that infects the insect. The kinds of resistance that we're interested in breeding are constitutive resistance and direct resistance. This is the easiest to screen for. Since you don't need to have the insect present, and you don't need yet another organism to assess whether or not you have beetle resistance. So this is some work that was done by our entomologist, Yvon Pelletier. We have set up at our center a field for a natural infestation of Colorado potato beetle. And in this field, the plants are put outside in trials to test for the beetle resistance. And he did this for about 30 different wild species and found quite a few that had resistance. And here they are here, Chromatophyllum, Puree, Brevicoli, which used to be Oclosense, Acroglossum, Possesectum, Tarihense, which is now also referred to as Bartolti. So these field resistance studies are really quite cumbersome to do, and they take a lot of time, and relying on natural infestation is always a little bit sketchy because the beetle might not emerge at the same time every year. And also the beetle has a tendency to be fickle, and if conditions aren't quite right, they might move on to someplace else. So getting consistent natural infestation is a challenge in scoring and phenotyping. So there is also indoor assays that are used, so Petri dish assays. But these Petri dish assays also have their challenges of their own. And I really wanted to point out a picture of my student Hannah McCoy here, and we this is also a story about how humans have had to adapt to COVID, and at the beginning of the pandemic we had limited access to the laboratory. So Hannah set up a beetle feeder arena in a closet at her house, and these are some of the results of her assay. And so in a sort of a Petri dish feeding assay, leaf discs are put into the Petri dish, and the amount of feeding is documented, and also the effects on the beetle. And you can see here that this is a choice and a no choice assay. So in the no choice assay, we have 2 pairs of tuberosum leaf discs in this dish, and then 2 pairs of Okidae, which is our wild species, in this dish. And then we compare the amount of feeding between dishes. And then we have a choice dish where there's 1 pair of tuberosum discs and a pair of Okidae discs. And we can look to see if there's any difference in choice. And here we see that indeed, the insects that are together with the tuberosum, they do a lot of feeding. There's a lot of leaf consumption, and also in the choice dish. But in the no choice dish, they simply don't eat the Okidae, and that's also reflected in the mass of the insect. So they gain mass in tuberosum, but very little when they're feeding on Okidae. And so one of the issues with doing these indoor assays is that the beetle is this sort of little brown insect. I don't know if you can see it that well on your screen, but the insect has this avoidance of Okidae. Sorry about that. I clicked something there. Here we go. Come back. All right. Here we go. So the insect has this behavior where it'll avoid eating something in the Petri dish, but when it avoids eating something in the petri dish, it has nothing else to do and nowhere else to go. And so what ends up happening is the beetle will starve itself to death when it doesn't have a choice. And so then you might score this as antibiosis because the beetle's dead. But in fact, it's still antizoonosis because the main driving factor for the insect is trying to avoid eating things it doesn't like. So there's a lot of difficulty in phenotyping Colorado potato beetle resistance. And that's something that we wanted to address, is to find a better way to score for resistance. And that comes from studying the interaction between the insect and the plant. So our goals really with screening these wild species is to do this introgression breeding. And introgression breeding for potato involves several different steps. And the main reason is because we're breeding a tetraploid plant where the wild species are diploids. And so we need to do a ploidy reduction on the tetraploid. And that happens with the use of a haploid inducer from another species, Solanum. So the haploid inducer will not contribute its genome in a cross. And so then the progeny that results is a 2X dihaploid whose genome is mostly drawn from the tuberosum. There's always a little bit of Freya that sneaks through, but this is mostly a dihaploid of tuberosum. This can then be crossed with the diploid wild species to create a 2X dihaploid diploid interspecies cross. And then this hybrid can then be used in the 2X introgression breeding. So I really appreciated Jaime Crohan's talk last week, talking about the introgression breeding that they do in eggplant, and that's such wonderful work. So the advantage we have in potato, it looks like we have more wild species that can introgress with potato. However, you know, I was really a little envious of Jaime as well because they don't have to deal with this sort of ploidy manipulation in order to get the introgression to happen and to bring it back into the breeding lines. So this sort of ability to use diploids in breeding is something that the potato breeding community is really sort of taking on. And one of the other areas of research that I'm involved in is diploid breeding and diploid inbreeding, and to try to turn potato into something more like tomato and an eggplant, where the breeding all happens with diploid inbred lines. So this is one aspect of breeding that can happen, is you can breed at the diploid level, but we still have so much material that's very adapted and has very good characteristics that's in the tetraploid breeding lines. And so we still need to do some of this sort of ploidy manipulation. So after you get it to a 2X interspecies hybrid, it needs to be converted into a 4X. And the 4X line is crossed then with the tuberosum. So once you use colchicine to double the diploid into a tetraploid, you can cross it with the tuberosum again to get 4X introgression lines. You can also take the interspecies hybrid that's at a diploid level and cross it with these meiotic mutants. The meiotic mutant carries a gene that causes it to make 2N gametes. That can then be crossed with the 4X tuberosum and get 4X introgression breeding lines. So this is all to say that the use of markers even in this step in breeding would be very helpful, and also the use of improved phenotyping would be very useful. So we've introgressed several different wild species in our breeding program, but today I'm going to focus on a discussion of the introgression of Solanum brevicali. We were very lucky in that the Solanum brevicali accession that we had carried in it tetraploid clones. So we didn't have to go through this elaborate ploidy manipulation process with brevicali, but this was a rare case. Most of the time, the wild species are diploids and you have to use this ploidy manipulation strategy. But in this case, with brevicali, we didn't have to. So in the cross with tuberosum, there was an F1 generation that was developed, and this was screened in the field for CPB resistance. And this was done by our breeder and the entomologist, David Ducoyer and Yvonne Peltier. And so the challenge that they faced was Fredericton does not make tubers under long days. And so the F1 lines had to be reared in a short day growth chamber to generate tubers, which were then planted in the field. So this was really a very laborious part of this crossing scheme. And then, of course, the ability to make tubers under long days was regenerated through back crossing with tuberosum over 5 back cross generations. And in back cross generation 5, there was another selection of only a small number of lines for CPB resistance by field defoliation. And the breeders have released 2 selections as varieties that carry this Fredericton and the CPB resistance. And you can see that after all this back crossing, the ability to make tubers has been restored. So, so there's a lot involved in introgression, and developing tools that can enable more efficient breeding is very important. So that's where I started in on this project, was to look at developing chemical phenotyping, which would be used in genetic mapping, and then use the genetic mapping to develop genotyping markers so that we can improve our capacity for selecting for CPB resistance using Solanum brevicoli. And so the introgression lines came from this from back cross generation 1 and back cross generation 2. And the lines from those 2 generations that had a variation resistance were used in the study. So as I said before, we were really interested in constitutive resistance, which meant that this was resistance that was there even without the insect being present. And the way that we examined this was to look at foliar metabolites using untargeted metabolomics. So we used leaves that were grown in the greenhouse without any Colorado potato beetle challenge, but we had plants that we knew came from a screening in the field for variation in Colorado potato beetle feeding. So we knew that the lines themselves carried these Colorado potato beetle resistance, even though we were growing in the greenhouse. And so this is where we started with our study. The untargeted metabolomics goes through a first step of liquid chromatography, separation of compounds by size, and then it goes into the mass spectrometer, which then gives us an accurate mass for the compound so that we can identify it. So the first study that we did actually was to look at some of the wild species themselves and to do a comparative analysis of metabolites between the wild species. And we looked at 6 of them and compared them to Solanum tuberosum. So these are the ion chromatograms that come from the liquid chromatography step. And you can see here with Solanum tuberosum, there's a large peak. This is the glycoalkaloid peak, and these are secondary metabolites that are toxic that are produced in Solanum tuberosum. They are the major secondary metabolite in the foliage of Solanum tuberosum. And you can see some of the other wild species also produce a large glycoalkaloid peak, but Solanum acroblossom and Solanum bertholtii do not produce glycoalkaloids. And we confirm this through doing the analysis of the mass spectra and deconvoluting the mass spectra. And we were able to determine that acroblossom and bertholtii do not produce glycoalkaloids. They produce other compounds instead. And so what we did was we compared what compounds were produced in the foliage of wild species with those in tuberosum. This was a fairly extensive study, but today I'm really just going to focus on what happened with the glycoalkaloids. So you can see already that tuberosum, TUB, here on the end, it produces a lot of solanine and chaconine. However, the other wild species don't really produce any of it. Some of the wild species, Chromatophyllum, brevicoli, Paucisectum, and puree, produce dihydrochromosonin. And brevicoli in particular produces a very high level of dihydrochromosonin. So this led us to focusing a bit on dihydrochromasonine as a possible source for a mechanism of resistance for the Colorado potato beetle. So what are steroidal glycoalkaloids exactly? Well, they're toxic secondary metabolites common to the Solanaceae family, and they're considered to be defense compounds for Solanaceae and constitutive defense compounds. So they're a good target for us to develop some kind of screening for Colorado potato beetle resistance. The chemical composition of a glycoalkaloid is a nitrogenous ring attached to a sterol, which is then connected to sugars. And the sterol comes from the biosynthetic pathway of cholesterol. And the glycoalkaloids are toxins for humans. A lot of them are. Some of them aren't so toxic. But solanine and chaconine are quite toxic, and their levels are regulated by governments. Now, the toxicity for a glycoalkaloid and a lot of the sterols comes from the fact that it has this steroid ring, and the steroid ring allows it to insert itself into membranes. And when it's inserted into a membrane, it can disrupt the membrane and disrupt the cellular structure. And so that's one mode of toxicity, and this is common to a lot of molecules that are steroids. The specific kind of toxicity that glycoalkaloids has is their ability to block acetylcholine esterase in synapses. So animals produce acetylcholine esterase in these synapses, and their job is to remove the neurotransmitter acetylcholine. So when an action potential hits, the acetylcholine gets released into the synapse, but you don't really want the acetylcholine to be there for a long time. You just want it to be there briefly so it can stimulate the muscle or stimulate the next neuron. Overstimulation is actually a very bad thing. And what glycoalkaloids do is that they block acetylcholine esterase, and then they lead to an overstimulation because of this excess acetylcholine. In fact, some pesticides target acetylcholine esterase. And interestingly, the Colorado potato beetle acetylcholine esterase is 150 times less sensitive to chaconine compared to other species. And it's also insensitive to pesticides that target acetylcholine esterase. So this sort of difference in acetylcholine esterase between us and the beetle is a possible route to developing targets for resistance in the host plant, in the potato host plant. So these are the foliar glycoalkaloids that we've detected in our studies on the wild species. And if we start with looking on the right side at the Colorado potato beetle hosts, Solanum rostratum, or the buffalo burr, is the original host, and it produces solanine. Solanum tuberosum, potato, produces solanine and chaconine. Tomato produces tomatine, and eggplant produces solamargine. So these are all glycoalkaloids the beetle can live with and consume just fine. In the species that the Colorado potato beetle avoids, there's Brevicoli, Chromatophyllum placysectum and puree that produce dihydrochromasonine. Chromatophyllum placysectum and puree produce also tomatine, neotomatine, but Solanum ochidae produces tomatine. So we think that in Solanum ochidae there is another mechanism of resistance that we're researching further. What's interesting is that the tetraose glycoalkaloids are less toxic to humans. So again, if you can find some contrasting toxicity between the beetle and us, this is a good target since you can increase levels to get insect toxicity, but that won't affect us. I also wanted to mention a study from Asaf Aharoni's lab. This is Cardenas et al. On Nature Communications in 2019. I think Dr. Szymanski talked about this work at his seminar. So here, as I said before, solanine and chaconine are glycoalkaloids that are toxic to us, but not to beetles. And here's another example of differential toxicity of glycoalkaloids. In the wild species Solanum chacoense, which has been used for a lot of CPP resistance breeding, it produces glycoalkaloids leptine, leptin 1 and leptin 2 that have acetylated glycoalkaloids. And these acetylated glycoalkaloids are toxic for the beetle, but not for us. The thing is that with the beetle has an incredible capacity for adaptation. And so although chaconine is used a lot for resistance breeding, we feel that there's a need to develop other sources of resistance to either stack or create variation resistance so we don't develop populations of resistant beetles. The other sort of downside of breeding for leptins is that leptins, the precursors for leptins, the pathway to produce leptins is shared with solanine and chaconine. And so to get high levels of leptins, you actually also have to boost high levels of solanine and chaconine. And this can be problematic since solanine and chaconine are toxic to us, but leptins are not. So there is some concern there in breeding for high leptins. Although the good thing about leptins is they're produced only in the foliage and not in the tubers. So this is sort of a good side. So going back to our Brevicali introgression lines, we did a lot of analysis on those lines. We did a complete untargeted metabolite analysis, but today I'm really going to focus on the discussion on the glycoalkaloids. So as I said before, we did a lot of CPB field defoliation studies. And so we were able to categorize the introgression lines as being highly defoliated by the Colorado potato beetle or low levels of defoliation. So we had high and low defoliation introgression lines. And then in those two groups of lines, we then looked to see whether or not there was any difference in the glycoalkaloids. And you can see that solanine and chaconine shows some amount of variation, but it's at a fairly low level. Solanitol chacotriose is a new glycoalkaloid that I'm going to talk about a little bit further that we discovered in our mass spec data. This had the opposite kind of effect from dihydrocarmosine. So dihydrocarmosine was higher in the plants that had low defoliation, but solanitol chacotriose was higher in the plants that had high defoliation. So it looked like they had opposing effects, where the solanitol chacotriose was associated with increased feeding, whereas dihydrocarmosine was associated with decreased feeding. So this was kind of a unexpected effect that we studied a little bit more. So the solanitol chacotriose, we looked at the mass spectra a little bit more. Solanitol chacotriose will focus on the two mass spectra and the opposite diagonal corners, and looking at this peak here, 414 for solanitol chacotriose, and the 398 is actually the A glycone for both solanine and chaconine, and the 414 is the A glycone for solanitol chacotriose. And what we were able to discern from the mass spectra was that solanitol chacotriose is an isomer of solanine with a modified A glycone. And there are other known isomers of solanine. This includes solamargine and beta-solamarine, which I'm not showing here, and also leptinin 1, which you'll remember is a precursor for leptins in the biosynthetic pathway for leptins. And some further analysis that we've done with HPLC has shown that solamargine and beta-solamarine are not solanitol chacotriose, but solanitol chacotriose could be leptinin, and we're still doing some research on that. So what we also found about solanitol chacotriose is that it's produced at high levels in some potato varieties, but not others. So Kennebec and Sheppardy produce solanitol chacotriose, but not Vinci and Russell Burbank. And also it's produced at very high levels in the foliage, but it's not really detectable in the tubers. So this is similar to leptin as well, which has production in foliage and not tubers. So we think that it's possible that it's leptinin, but we are still doing some additional structural analysis on the compound. So The Solanine chacotrios had an opposing relationship with dihydrochromosonin, which is very interesting. So they were, where Solanine chacotrios is high, dihydrochromosonin is low, and the opposite is true, where dihydrochromosonin is high, Solanine chacotrios is low. And they also had opposing effects on the beetle. Solanine chacotrios was associated with feeding, and dihydrochromosonin was associated with deterrence of the beetle. So this indicated to us that the beetle may be sensing both of the compounds, and the compounds may have a shared pathway where the precursors may be used for producing one pathway that leads to solanine chacotrios, or they can be used for dihydrochromosonin, but not both. This is something that requires a little bit more research, but I think what we can conclude from this is that the beetle is sensing not only levels of dihydrochromosonin, but also levels of a compound that it likes, which is the solanine chacotrios. We know this also because when we have plants that don't produce solanine chacotrios that have introgressed with Solanum brevicoli, the dihydrochromosonin levels alone do not produce as much contrast in Colorado potato beetle feeding as those that have opposing levels with the solanine chacotrios. So we wanted to examine this sort of this contrasting relationship and also the sort of ratio difference for solanine chacotrios to dihydrochromosonin. And we did this using a method called selective genotyping, which is a version of bulk segregant analysis. So in bulk segregant analysis, you would physically pool together DNA and then sequence that in a single run. In this method, we actually did genotyping by sequencing for all the individuals in the population, and then in silico pooled individuals that had extremes in the solanine chacotrios to dihydrochromosonin ratio. So we calculated the solanine chacotrios to dihydrochromosonin ratios, identified those that were in the extreme, and then we looked at the single nucleotide polymorphisms in these extreme populations to identify loci that were associated with controlling the ratio of solanine chacotrios to dihydrochromosonin. And we found a region on chromosome 1. And it's a fairly large region, but within that region, there were several genes that were possible candidates involved in controlling the synthesis of either solanine chacotrios or dihydrochromosonin. And interestingly, game 32, which was from the Cardenas study, was also found on chromosome 1 for at least one of the lines of chaconine. So this is a region that we're pursuing a little bit further for development of genetic markers to select for favorable solanine chacotrios to dihydrochromosonin ratios, and for the purpose of finding clones that are resistant to the beetle. So I'd like to come back a bit to talking about this, the studies that we had on the ratio, the glycoalkaloids, and sort of come back to this sort of finding that almost all of the wild species that had Colorado potato beetle resistance did not produce solanine and chaconine. And the question that we had was, do solanine and chaconine provide benefits to Colorado potato beetle? Are they sustaining them in some way? And so actually reducing them may have some effects as well on the Colorado potato beetle. So we collaborated with Ashraf Al-Haroni and Max Itkin at the Weizmann Institute to do this work. They had an RNAi construct to silence the glycoalkaloid metabolic enzyme 4, the GAIN4 gene. We use this construct to transform Binchi, a potato Binchi variety. And we used Binchi because it did not produce solanine chacotrios. And so we were able to really focus on the other glycoalkaloids, solanine and chaconine. So the GAIN4 encodes P450 enzyme, and this functions in the steroidal glycoalkaloid biosynthetic pathway. And the work here that I'm talking about was mostly done by my postdoc, Dr. Jamuna Padel. And Jamuna found that the RNAi plants did produce lower levels of gain-for transcripts. And this was accompanied by decreased levels of steroidal alkaloids and steroidal glycoalkaloids, and also increased levels of steroidal saponins, which was similar to the findings that were reported by Maxkin et al. In Science in 2013. What Jamuna also did was she looked through the mass spectra and found evidence for increased levels of phytoecdysteroids as well. And so phytoecdysteroids are very interesting because they mimic an insect hormone, ecdysone. And increasing the consumption of phytoecdysteroids may also have effects on beetle development. So there was not only a reduction in glycoalkaloids, but also an increase in other steroidal molecules in game-for RNAi plants that could affect the beetle. So in looking at the foliar glycoalkaloids, we found that two lines that we were going to pursue in the rest of our studies, which were T8 and T9. And T8 and T9 had slight differences between them. T8 had low levels of both chaconine and solanine, whereas T9 had low levels of chaconine, but its levels of solanine were moderate and higher than they were for T8. So when we looked at the survival rates of beetles feeding on game-for RNAi plants versus beetles feeding on wild-type plants, we found that there was an increased death rate amongst beetles feeding on the game-for RNAi plants. And so this also supported our hypothesis that these glycoalkaloids did do something to support the beetles. So that was one sort of evidence that glycoalkaloids are not always toxic. They can also provide beneficial supporting factors for the beetle. Jamuna also did a developmental analysis of beetles that were feeding on the game-for RNAi plants. And what she found was that there was a shift in the development. So every dot here is an individual beetle. And here you can see the different life stages of the beetle, like larval stage 1, 2, 3, and 4. I've left out the pupa and skipped to the adult, but the pupa more or less looks like a cocoon and it's in the soil. So the different life stages are different colors. And so each dot is an individual beetle and its life stage is indicated by its color. And in this diagram, we see that in the wild type plants, there's a fairly extensive range for the L4s and for the pupa. Whereas this exchange of this sort of purple and pink is a little less for beetles that have been feeding on the game-for RNAi plants. And so overall, the life cycle was shortened. So the developmental stages were shortened for beetles that were feeding on the RNAi plant. And but the beetles still consume the same amount of leaf and also gain the same amount of weight. So their feeding increased, when they were feeding on these game-for RNAi plants. And in addition, most of the pupal stage was sort of shortened, or the shortening of the lifespan or shortening of the life cycle was mostly in the pupal stage where the beetles aren't feeding and they're sort of pupating underground. So we attributed at least some of this difference in development to the increased phytoecdysteroids the beetle was consuming, but it may also have something to do with the decreased levels of the glycoalkaloids. We also looked at the Colorado potato beetle acetylcholine esterase and activity to see if that was affected by the extract. So not only the extracts of the glycoalkaloid, but the entire extract, entire plant extract. And so, as I said before, the T8 plants had low levels of both chaconine and solanine, whereas the T9 plants had only decreased levels of chaconine. And we compared the extracts from these three different plants and their effects on the acetylcholine esterase activity of the beetle. And what we find is that the T8 and T9 had lower levels of acetylcholinesterase activity than T8. So usually glycoalkaloids inhibit acetylcholinesterase. And so if there's increased activity of acetylcholinesterase, then that means that there may be a loss of inhibitors in the plant extract from T8. And that's how we interpret this result. So we're continuing to do some more work on the effects of consuming low glycoalkaloids in the beetles. We have some hypotheses that we're working on. First of all, in beetles that are consuming wild-type plants, these wild-type plants have very high levels of glycoalkaloids. So the beetle has adapted to feeding on high levels of solanine and chaconine. And so what we think happens in the beetle is that it has this very strong acetylcholinesterase activity that's resistant to glycoalkaloids. And having that kind of high level of acetylcholinesterase activities prevents the inhibition by glycoalkaloids and also prevents this overstimulation by acetylcholine. When the beetles switch to feeding on T8 plants, which are low in solanine and chaconine, the glycoalkaloid levels are lower. The inhibition on the acetylcholinesterase is no longer there. However, the beetle has already adapted and has this very high level of acetylcholinesterase activity. And then for that reason, it then reduces the level of acetylcholine and to levels that alter the neurotransmission and reduce neurotransmission. And indeed, Jamuna observed slower movement and altered behavior of the T8 gainful RNAi plants. So this is work that we're continuing with in the laboratory. I just want to summarize now and summarize the talk. So we found that the wild Solanum species, Solanum brevicale, Puree, Chromatophyllum, Posisectum, Acroglossum, and Bertolti had CPB deterrence and were useful for introgression of CPB resistance. There was an intercross done with Solanum brevicale and Solanum tuberosum to introgress the CPB resistance genes from brevicale. We have now investigated further the nature of this resistance from brevicale to develop improved screening methods for this kind of resistance. We found that dihydrochromosonin is a glycoalkaloid associated with deterrence, which has opposing relationship to solanine alchapatriose, which is a glycoalkaloid associated with feeding increase. So looking for a QTL that is associated with the ratio of these two compounds was done, and and we now have candidate markers to select for a ratio of those two glycoalkaloids. We also demonstrated in a gain for RNA9 knockdown plant that solanine and chaconine are important for sustaining the beetle, and this may be related also to the increased levels of ecdysteroids when glycoalkaloid levels are low. So I'd like to end by acknowledging the many people that have contributed to this work in my lab. Jamuna did the work on the genetic mapping and the gain for RNAi. Craig Worrell was a student that worked on the wild Solanum on metabolomics. Hannah is my current student that's working on Solanum okidae, and Sui Jin is our analytical chemist, and Emily is the new technician in my lab, also working on okidae. I'd especially like to thank Yvonne Pelletier, who is our retired entomologist who did a lot of really important work on identifying wild Solanum with CPB resistance. And Kyle Gardner worked with me on the genetic mapping, and David DeCoyer and Benoit Bizimungu and Agnes Murphy were the breeders that worked on developing the introgression lines with brevicale, and Jun Song and Leslie Campbell worked with me on the metabolomics, as did Larry Calhoun. And a very big thank you to Asaf Aharoni and Max Itkin at the Weitzman Institute for sharing their gain for RNAi construct. So yes, thank you.
Awesome. Thank you so much. So now we're open for questions. So if you have a question, you can either type it in the chat and I can read it out loud, or you can just type that you have a question. Stacy has a question. So do you want to start us off, Stacy?
Oh, hi, Helen. Thanks for the talk.
Thanks.
That was fun. I'm aware of the Colorado potato beetle, but I was really fascinated by that history, the story of like coming up with Solanum rostratum first and then diversifying in what it ate. And I guess I'd been wondering this whole time about that. So is there variation in the Colorado potato beetle still? For like, are there still specialist populations out there, or basically has this crazy eat the heck out of things version spread around the world and that's now the dominant type of potato beetle that you find?
That's a really great question, Stacy. And there's been a lot of work done on Colorado potato beetle populations, and I think that the population is fairly diverse, and the specialization still is not really that distinct yet. What we are seeing are isolation of populations that are resistant to the pesticide. So there are these pockets of pesticide-resistant populations, but even within those populations, there's still a fairly large amount of genetic diversity. So that's one of the characteristics of the population overall is that each even sort of geographically isolated population is still fairly diverse and still carries a lot of capacity to adapt. But there are certain genes that people are able to identify now in resistant populations, but the population that sort of originally specialized on rostratum, it still remains fairly diverse and able to adapt to other hosts.
So I guess just a follow-up from that is like, how do you think that's going to complicate the breeding efforts if there's all this genetic diversity in the beetle that's kind of could compete with strategies that are produced to fend it off?
That's an excellent question, and I think that this is sometimes something that we debate with the entomologists. I think at the end of his career, Yvon said that he didn't really think that there was a way that we could breed for resistance, because he did work originally with Bertolti, and they had a successful introgression, but the field trials showed that the beetle adapted to that within about 2 or 3 years. So I think that he started to diversify even more Solanum. And so now, our thinking is that we need to think of it like late blight, which is a pathogen that's very destructive for the potato, and stack resistance. So a lot of these people that breed for late blight resistance will stack our genes. And so they've identified this group of genes that they can combine together and use a combination of our genes to defend the plant. And that's what I'm thinking also for Colorado potato beetle, is to have a combination of metabolic pathways that produce different metabolites in the foliage, and also to integrate our breeding strategy with integrated pest management strategies. So knowing, for example, that Solanidinol chacotrios is something of a feeding attractant and a phagostimulant for the beetle, we could plant around a field these kinds of plants, or create a trap plot with plants that have the Solanidinol chacotrios, and then attract the beetle there so that they will feed on those plants. And then at the same time, your resistant plants may have lower levels of that metabolite together with some resistance metabolites. So develop strategies for cultivation that will prevent the development of resistance. And that's really a very big challenge for commercial producers. But I think that if you're really going to develop strategies that prevent these cycles of infestation and overuse of pesticides, something has to be done to integrate breeding with integrated pest management.
Awesome. So I kind of have a question. I don't know if this is a really naive question, but it's kind of like from the plant perspective, because it seems like the plants are just producing these chemicals and these steroids statically, correct? And like you were able to manipulate not to do it. But is there any like regulation of like, okay, I'm getting eaten, so maybe it upregulates some chemical compounds? And do some species do that better than others? And what does that look like?
Yeah, that's a really good question. And I think that there are, within a single plant, there are multiple strategies for resistance. And the constitutive defense strategy is one of them. And the beetle does, I mean, the host plant does try to produce other mechanisms of resistance to the beetle. And sometimes we see this as production of other kinds of compounds. And so some of the other compounds that we see are polyphenolics that are also used in defense. So there's other defense metabolites. And the beetle may, the plant may also sort of change their cell walls and produce more sort of defensive structures that way to make themselves a little bit more difficult to digest. And the plant also responds to being fed on and changes its own sort of developmental pattern so that it, diverts more resources towards recovery. And so some of the regurgitants or saliva can stimulate some of these changes in the plant, even without being chewed on. And injury alone can stimulate the plant as well. So there's a lot of different layers in which the plant can respond after being fed. And you're right, the constitutive defense is really just one aspect of its defense.
Does anybody else have a question? Oh, Federico, you can go ahead. You can unmute yourself.
Hi, yeah, great talk. I have actually two questions. The first one is regarding the strategy to generate like a defense that is that can last for a long time. So you mentioned that with argines, kind of the new method will be to stack different argines. And my question is, so I think secondary metabolites and toxins have not been so much used to generate improved varieties that are more resistant. But do you think first that secondary metabolites provide longer resistance than argines or not? And second, do you think that it's possible to stack the plant with toxins, like different ones, just to make sure that that is better? Or it's very likely that it's going to be toxic for the plant, or that there's going to be like some pathway loops that will prevent from producing all of these toxins?
Yeah, that's a great question. And yeah, I think that, argines are very attractive. And the thing with insects is there's not always an argine, although some people are looking into these sorts of, effector related pathways that are triggered with insect feeding. So that is an aspect of resistance that can be there. And that's probably easier to stack than a multiple metabolite-based strategy. Because if and I think that you mentioned this as well, is that if you have a lot of different metabolites going on, it can actually create a mixture of biosynthetic pathways with unexpected consequences. And also it can be very taxing on the plant's resources. And so, it might reduce our tuber yield, which you don't want to have as well. Maybe a way to tackle some of this resistance that can develop is again coming back to not planting monocultures quite as much. So you might have some potato varieties that produce one kind of metabolite defense, and then you plant that together with potatoes that have another kind of metabolite defense. So maybe not stacking the different metabolites together in one plant, but having a planting strategy that mixes things up so that you don't make the field as prone to Colorado potato beetle resistance development.
Good. And my other question was kind of a more genetic, philosophical one. And I'm not sure if the region that you identified in chromosome 1 has a SGA cluster. Does it? But so when we have a cluster, sometimes as geneticists, we always think, okay, there should be one gene there that is important. But actually, there might be them all, alleles in all, or at least in many of them. And it's not a single gene, but all of them. So
yes.
Do you consider that? How would you test it? And
yeah, that's a really good question. And I think that this is my chance to say that the region that we identified in chromosome 1, our SNP markers were really far apart. So we really need to do some more fine mapping to get it down to a region. But other people have identified this region as being associated with glycoalkaloid production. And I think that what we see in these regions is not just one. Like in R genes, you see a whole bunch of R genes, but in some of these clusters or gene regions that are associated with glycoalkaloids, you see more than one enzyme that's in there. Maybe not the entire pathway, but there's sometimes a cluster of enzymes that are related to each other in a biosynthetic pathway. Or you can also see in there transcription factors that are related to controlling biosynthetic pathways as well. So I think that, what we'd really like to do is use some RNA, use some, genome editing type strategies to target some of these genes that may be associated with biosynthetic pathways. And the advantage there is that we can go and chemically analyze the plant to see whether or not the pathway is actually disrupted and changed. So, so some gene editing or genetic modification strategies, once we've narrowed down to a certain number of candidate genes, is feasible.
Thank you very much.
Thank you.
Awesome. That might be all the questions. If somebody wants to, they can chime in. I'm sure you can reach Helen Tai via email if you have questions, if you're watching this later as well. Next week's going to be a little bit of a different week. Luke might be able to say more about this.
Well, yeah, I was gonna ask everyone about this because I realized it's a holiday here in the US next week. A lot of people probably have that Friday off. So the speaker for Friday canceled. So we were thinking of doing some sort of other activity. I don't know, be interested to hear everyone's thoughts, especially if there are going to be a lot of people who won't potentially even be able to come.
So we're thinking about doing a discussion, and we'll definitely maybe send out a poll on Twitter about different options that we could do if people are interested, either doing a little short quiz just to build community or doing more of a discussion-based things. So maybe we'll look out for Twitter and maybe be active on that poll. That would be actually super helpful. And at least by Wednesday, we'll send out an email letting everyone know what we're planning on doing. Thanks again, Helen, for giving your talk. It's really interesting. You've done so much stuff, and it's really great to hear your research.
Well, thank you very much for inviting me, and thank you for organizing the community in this way. It's been great.
Awesome.
Thanks.
Have a good week.
Bye, everyone.